Very good morning, students. Welcome back to lecture 3 of the course Immunology and Immunotechnology. Today I will be covering evolution of immune system and innate immunity for the lecture for 30 minutes or so. So the lecture is divided into several topics where you can better understand the concepts. So I just divided the lecture into 6 modules or 5 modules that cover up each module will have some similar topics we have to combine and discuss. The flow of the lecture will be, let's start with the basic understanding of evolution. We can brush up something on basic aspects of evolution there and then go up into how the immune system has evolved so far. So go ahead with the immune system evolution. A little bit more concepts on innate immunity and adaptive immunity. We can work on that. And then we can go a little bit on basics of immune response that we already discussed in the class 1 and 2, little bit more memory. Remember, you can bring back your memory on immune response, a little bit more on the concepts of innate and adaptive immunity and the process steps involved in immune response. And we go in more detail on innate immunity, inflammation, and toll-like receptors. What are the receptors? How they are going to help in the immune system and the recognition process. So that's a lecture plan for the class today. Let's go ahead with discussing evolution. One of my favorite topic, evolution. So we all know that we evolved from primitive organisms, a single cell, to multicellular organisms. And the primitiveness becomes through evolution, a complexity increases. For example, of what we have studied in terms of, for example, if we take an example of evolution of the eye, from primitive to complex organism, from unicellular to multicellular organisms. So there seems to be the primitive eye. There will be a primitive eye that is formed, and it has gone through some evolutionary processes. For example, the system itself, the development of the eyes itself will work through an evolutionary scale of millions and billions of years. It adapts itself to better suit to the environment, to the conditions where it can survival of the fittest for the Darwinian system, Darwinian concept. So it started with single cell, some primitive eye, then it matured into the receptor development, light recognizing development. Then it went into having the retina and the light capturing system and the lens. Many advanced organs have developed. For example, you can take an example of eye development. But the same way you can limb development, primitive limb, you can look into that. And also primitive reproductive system development. There is a very beautiful examples, review articles go through that will help you understand the evolution in a better way. And also we know that we have a proof of concept that we are evolving and we have a rudimentary or vestigial organs, for example, rudimentary tail, body hair, wisdom teeth. This all gives an example that we have the part of the system in our body are evolving constantly and have some remnants for the vestigial organs, we call them. So another aspect, evolution proof is from fossils. We study to fossils from dinosaurs to aphids, the honey sucking aphids. All those you can study, and one of the concept we clearly mention in evolutionary studies is homology. You have homologous organs, you have homologous systems, homologous genes that helps in studying a several concepts, specific concept that evolved through the system. For example, a question we are asking here is in which organisms eyes appeared first? Because we know that unicellular, we know that some of these our ancestors lived in the sea only. When they are in the sea, they don't need eyes. So the eyes may be primitive in that form, but where it formed and how it formed is a general question of evolution. But coming on to, in terms of homology, I discussed again, evolutionists tend to focus on similarities. For example, you take a formation of the limb development in humans, you compare it to the lower organisms, you can see a similarity in that. There is a structure, 3 structural units of the hand. One example, digits here, tibia, femur, all of them have one unit that is kind of, you can see the unit has been conserved all along the evolution. And then there is a morphological changes depending on suit of functionality, but overall you can see a similarity. That's homologous organ, you study with that. So that is in general of evolution. But coming on to immune system, we already studied the immune response, right? We already studied in the last class level of protection in terms of any organism. There will be a level of protection in terms of physical barriers and also in terms of later the innate immunity kicks in and then acquired immunity comes in. So this we discussed in the last class. It's a source is NPTEL Biotechnology course. The graph represents from the course material and the level of protection in terms of physical barrier and the immune responses. How quickly an immune system works on, innate immunity very quickly works on, minutes to hours. Then it can work through. If the system efficiently works, it will take off the pathogen and it will give protection to us. At the same time, acquired immunity, that shows it takes some delayed response. It works through a week. So I just want you to look in this graph that we already know and look back into the evolutionary perspective. Let's say you take off acquired immunity out. You are going in a timeline here in a terms of evolutionary scale, starting from millions or billions of years back. This is the, if you take the graph here, this is the present day. We know that present, what are the immune systems as a whole organisms we have. But if you take it back, go to the primitive time, go back in past, you start with the primitive system, primitive immune cells. One thing I can see that I can take off acquired immunity, I can take off innate immunity also, but I can really look into it as a basic concepts. As a non-specialist, I can look into only a physical barrier. Yes, we know there are amoeba, there is hydra, there is several animal phyla, arthropods, insects, everything are there. I can think that there is a protection from an infection, from an attack from a foreign body or foreign animal. That one thing is skin barrier I can think of. Then in the evolutionary scale, there will maybe primitive innate immune responses, primitive chemicals produced. Usually in the olden times, all this is chemistry only. The bacteria produces some chemical compounds. The other, when it attacks some organism, the organism produces some other chemicals, all chemistry, right, at that time. Then the chemistry evolved into a complexity. The complexity goes into cells and organisms. Then T lymphocytes, B lymphocytes, macrophages are formed here in terms of innate, then go back into getting into T cells and acquired immunity. So there seems to be a complexity of the immune system that has evolved during time, starting from simple chemical molecules. And it's why the complexity is needed, because the environment is changing, the organisms are changing, the attacking agents, disease causing agents, they are evolving through. So the immune system should be ready to defend them back. So they have evolved to starting from primitive cells and chemistry, chemical molecules produced, to that innate and acquired immunity. That is one of the concept of evolutionary aspects of immune system. You can ask, you can put two questions for that. One question is, for example, you are looking like here, the evolution of an immune system, post-pathogen interaction and co-evolutionary concept. The question I can ask is, in which organism first immune cells appeared? What was the first defense mechanism or defense molecule that's been observed? I can think of, I can look into that. So there was a lot of studies have been done on immune system evolution. And there is also two concepts in evolution that you need to understand. One is convergent evolution and divergent evolution. We don't want to go in deep into that, but I will give an example of one of the immune system development, one of the studies done. So here I'm in this slide. It's a big slide, but bear with me. I will guide you through the slide, and then to the right information, you can go and understand the slide in better way. The slide, we adapted it from Dr. Gus Enhart from University of Toronto. So he explains the concept of convergent evolution of the adaptive immune system. So he's talking about adaptive immune system. He's talking about a concept of convergent evolution, and he's linking antigen lymphocyte interaction in two organisms called Jawless and jawed vertebrates. It's okay. So this is the heading we are discuss in this slide. But I would like to brush up your memory towards your classification, zoological classification. Go back into phyla, different animal phyla. First, start from in the middle here. You look through that. So this straight line up towards that represents an evolutionary scale moving up with the time, starting with the phyla Cnidaria. It goes through arthropods, nematodes, molluscans, Annelida, Echinodermata, Urochordata, Cephalochordata, and again it branches out into two organisms. One is Eneta and Gnathostomata. Eneta are jawless vertebrates, Gnathostomata are jawed vertebrates, including humans. So this is the animal phyla. And you look through here, the S-shaped going downwards, it explains the concept of immune system. This is a interaction between antigen lymphocytes, where you start with 1, 2, 3, 4 steps here. First step is detection and binding of free-floating antigen. We already discussed that antigen-antibody interaction, antigen-presenting cells, how the cells interact with the foreign molecules. And then a specific concept, endocytosis. The third one is presentation of processed antigen to the cell surface, and then antibody release. These 4 steps. This is concept of how we are responding to the attack of a foreign. So look into that here, this arrow, this S-shaped downward arrow. Look into 2 organisms in evolution. One is Aenata, one is Gnathostomata. Let's take ourselves, humans, vertebrates. We already know that B cells, T cells are part of our immune system. The first step, recognition of the immune molecule, happens with the B cell. Antigen comes and attacks to the B cell. Then B cell takes through it endocytosis, and then it processes the antigen to the cell surface. Then the secondary immune responses that T cells will take up, and the specific response will be elicited. According to that, specific antibodies will be produced, conventional antibody production. So this is vertebrates. Let's go back in evolution in Aenata. Is there a homologous system there? People were trying to, they were interested. This is my system. If I go back to Aenata, do I have a homologous similar system or not? Yes, absolutely. The mechanism looks, all the steps are common. 1, 2, 3, 4 are common to the on the right side. But there is a change in the structure or change in the molecular pattern that whatever recognizes that. For example, it's a B cell we call here, we all call VLR B cell. Instead of T cell, we call it as VLR A cells, and the process is same, but mainly look into the antibody production here. This antibody is biforked, right? Bilobed antibodies here, that's in humans. But here you see that it's a octameric, I think, octameric or decameric antibodies here. That's circular and having more lobes and more bodies. Here only 2 antibodies are there. So that gives the antibody specificity and recognition capacity. So in summary, this slide talks about evolutionary concept of the immune system in 2 species. It talks that there is a conserved mechanism of steps involved in that, but there is a difference in the production or structure of the antibodies produced, differential recognizing cells, but there is a consistent homology within the system. So this is the description that talks about more in the previous slide. You can go back to the text and read it that, but I would like to credit this information to again Dr. Go Sinha from the University of Toronto. So that we talked about adaptive system here, right? We're talking about adaptive system, how it is consistent, conserved. Let's go back a little bit more into evolution. Let's go back into 700 million years back here, and we are here now. So you are looking in the concepts of here. At that time, we know that only sponges existed. Then you go back 450 million years. This MY is million years abbreviation here. You have fishes, then you move up, you have as humans. So in terms of evolution of the system, physical barrier, innate immunity, and adaptive immunity that evolved so far here. So I would like to focus on this graph or this portion of the slide, where you see that in the primitive systems where sponges are there is a specific responses, very quick and efficient responses that don't take much time. So the mechanisms were called as antimicrobial peptides. We know that AMPs, and one of the phagocytosis that I will discuss in the class further. And then complement system, toll-like receptor. Complement system we will talk in the next class, but toll-like receptor also I will try to cover today in this class. So at that primitive level, sponges will have these 4 systems that has been placed to defend the, to elicit an immune response. When move up into this 50 million years, when the fishes are there is specific compounds, for example, lymphocytes are developed, major histocompatibility complex appeared, and then immunoglobulins, antibodies, and then T cell recognition and few other concepts evolved. And move up into that, we have more complex systems here that we will discuss in the further class. But I would like to keep an eye again, evolution. There is a system of evolution that has placed from the, could someone please mute your microphone? Thank you. So the system evolved accordingly. So again, this text will guide you to go through that information presented in the previous slide. So let's talk in this slide about stages of immune response that we are already speaking from past 2 classes. There is a recognition process, antigen presenting cells, and then a processed molecule, then antibodies and T lymphocyte. You move up into further steps. For example, here infection, or here is the recognition, the effector response that the cell can make up. So basically the immune response is divided into 4 stages: recognition phase, proliferation phase, response phase. Just imagine that you're shaking a hand with bacteria or a virus. Okay, in a case, an example take here, and then that is your first step. You are in a contact with the foreign pathogen. That is, how do you recognize the foreign pathogen? That step, antigen presenting cells in our body, they take from the shake hand, they know that you are touching a foreign object. They will start their response. They will look into that. They will recognize the molecule self or non-self. Then if it is a non-self, they think it's a threat to the body. They want to go and elicit an immune response that is going to response and the effector stages. But before that, they will go to a proliferation phase. For example, they have to produce based on the how many pathogens or volume of the pathogen that's entering into your body. First they recognize, then that's a cognitive step, recognition phase. Then they go up into a proliferation phase, where specific molecules that are going to defend the initial molecule, that is touch antigen, and the antigen, any immune response defective or any foreign poisonous material or any destructive material that gives, they want to defend it. So they want to go in a volume based. They can go for proliferation stage, more lymphokinases, where lymphocytes are produced, then response and effector phase. Separate cells have been activated. Cells are activated, then you go up into effector phase. Okay, effector phase is either you go ahead with making any tissue damage happens, that you can respond to that, or you can say a lysis infected cells, clean up of the lysis. Okay, that's clear to all stages of immune response. Okay, I'm moving to the next slide. So innate immunity. In terms of innate and adaptive immunity, we talk about, let's take functions of the innate immunity in a summary slide. We know that so far, innate immunity is defend and eliminate infection at entry point. It helps in tissue repair and elimination of infectious cells. So innate and adaptive immune responses work together in concert to elicit an immune response. So we already talked in previous classes that innate and adaptive immunity in terms of bacterial response or extracellular, intracellular agents. The one thing we have to recognize, we spoke about PAMs, DAMs. DAMs are damage and associated molecular particles. PAMs are pathogen associated molecular patterns. So we call them dangers. We call them as dangers. Innate immune takes control of the both of them, and then it provides a specific responses here. Immune cell that we already discussed. Tissue repair, inflammation, adaptive immunity, cytokine, chemokine. Okay. So let's take an example of inflammation in body. For example, when inflammation is that, so you have a bacterial inflammation or a tissue repair, the cells or tissue gets inflamed. A kind of, for example, heightened fever is also an inflammation response, inflammatory response in the body, that you are having some kind of issue with the body, so you have to defend. So this material is coming from Immunology Cube, 7th edition. In a quick response, I will talk to that. A tissue is damaged by some process, then along with that, some bacteria entered into the system. Okay. So first, tissue damage, local response molecule, there is a recognition process that tissue is damaged. Then immediately through blood capillaries, where the process has to be repaired, the capillaries will allow permeable influx and fluid of cells into the where attack of the spot. Then the cells will come up, a process called extravasation will happen, and then specific neutrophils and other phagocytes, which are part of the immune system flowing through the blood, will be released at the site. In one of that, the yellow cell we are looking here is a neutrophil. The neutrophil will go into two processes, either chemotaxis or phagocytosis, that recognize the molecules. Then it will take through a specific phagocytosis mechanism and kill the molecule. This is one of the inflammatory basic. And this is one of also part of the innate immune response. But if you take in another concept, the phagocytosis here, it's one of that simple, again, another mechanism where innate immunity depends. Phagocytosis. Bacteria recognition happens, bacteria is ingested, and a specific phagosome will form. Along with the phagosome, there is again intracellular mechanism that allows phagosomes to lysozyme. Lysozyme again produces lysosomal enzymes, kills the molecule, particle goes up. This is also very simplest mechanism of phagocytosis. I wish we had only this mechanism. We should not be an evolved so far, so that only take the bacterium, kill it, that's done deal. I'm out of my sickness. But why the immune system evolved? Because the bacteria and viruses are so smart. They evolved, they evaded the system of phagocytosis. That's why we are going into some complex mechanism of tissues that inflammation is also required. In primary and secondary immune response, we will talk about it more in terms of vaccination and antigen-antibody interaction and in kinetics of antibody production in the future classes. But again, first infection, it will have a lag phase in the process. Then infection will produce an antibodies here in the blood. Second infection, B and T cell memories. There is no lag phase here. The quick response will comes up. You can produce a lot of antibodies within the time. You can go ahead with the text here. So we are running out of time. I will try to cover the remaining slides. The primary response is slow. Secondary response is prompt and powerful. And you should remember the terms primary and secondary. So again, we talked one concept in the previous classes that how cells recognize as antigen presenting cells will recognize an antigen and then antibody antigen interaction, ligand receptor interaction. So this slide will talk this material further reference, further additional material for the lecture. You go to this article, you go ahead with studying the process where NK cells will have a specific CD16 receptor, how they respond to infected virus cells. You go through and read this material. I may not cover this slide due to time constraint. I will go ahead with the next slide where we are talking more about that toll-like receptors. One of the concepts we looked into the primitive sponges that we have seen there is a mechanism of toll-like receptor. The toll-like receptors are part of the immune system, innate, and they are also majority, principally they work with the innate cells, but they may work with an adaptive and a non-immune cells also. They are specific molecules, they have TLR4 receptors, the specific structure of the molecule that I will talk in the next slide. They have a specific recognition pattern. They sense tissue damage, because these are the receptors for immune system. They help the immune cells to recognize tissue damage. Later, according to that initial response, they will give an immune response and try to repair the tissues. In evolutionary perspective, again, toll-like receptors, they evolved in recent times. They have been there 700 million years back. For example, RNA viruses, DNA viruses, there is toll-like receptor that it's there in the gram-positive, gram-negative, and fungi and proteins. These receptors on cells help the immune system build that. So there is specific phylogenetical profile. You can look into that. Okay. Toll-like receptor are a family of transmembrane receptors that serve as signaling receptors in the innate immune system. Their ligation by exogenous and possibly endogenous ligands triggers a pro-inflammatory and then inflammatory response that helps the immune system defend it. If you go back into TLR structure and function, this is the cell membrane. The TLR has extracellular, intracellular membranes. Extracellular is leucine-rich repeat. Triactivated lipoproteins, lipopeptides is one of the function, and there may be a transmembrane helix, that's the structure in the cell membrane. It embeds into the cell membrane. An intracellular domain, it's called TR domain, it is a heterodimer. We will study a concept in future in further classes that how these toll-like receptors work in illicit response at the molecular level. We will study in depth what happens above, what happens below on the cell membranes. That all we will cover in the future. You can read a little bit more about toll-like receptors here. So, okay, so far in this class, I covered basic of evolutionary aspects, basic of how the evolution of our immune system has come up, what is the necessity, what is the requirement. And further, we spoke a little bit about innate immune system, what are the functions and the different mechanism, how innate immune system will defend us against pathogens, and then a specific toll-like receptors and of inflammatory process and phagocytosis we talked about. Okay, in summary, last slide, I will talk about synergy between adaptive and innate immunity because they work in cohort. The innate immune system has to be active that can work in the first recognition steps. Then it can elicit a response for the adaptive immune system. So both have to work in concert. For example, let's take an infection, the dead end. The result is that destruction of the pathogen or any tissue repair you have to do. How it's been done? The mechanism starts with innate immunity. Innate immunity responds to either intracellular, for example, stress in the body, or extracellular here through a specific immune cells, NK natural killer cell, phagocytes. They go through a mechanism and destruct the pathogen. At the same time, if this is not enough, if the molecules are smart enough, or the pathogens are smart enough, then the innate immune system goes into an alternate pathway of adaptive immunity, where again specific cells of the immune system, T cells, B cells, will come up and antibodies antigen interaction elicits. Again, there is adaptive immunity extra and intracellular and cellular specific cells will come up. Again, at the end, they will go ahead with destruction of the pathogen. Okay, so just go ahead through the slide. I will conclude the presentation giving the time limit. We will bring back tomorrow, probably afternoon. I will talk about acquired immunity a little bit more detail. So try to understand that these initial classes will be of introductory only. More advanced concepts and materials will be given in the further classes. Okay, have a nice day. Thank you.